We'll try to finish the female reproductive today, but we'll see how far we could get into it. Okay. Okay. First off, as we're developing, we have homologous structures, meaning that these structures are very similar to both sexes, and they have similar functions. And examples of these are like the gonads themselves. The gonads are our primary sex organs that will produce our gametes. And what are gametes again? Are what? Sex cells. So for example, for females, what is our sex cell? The oocyte or ovum, right? Eventually the ovum. And then for males, sperm. Very good. And then also sex hormones. It produces sex hormones as well. So for females, they are the ovaries. For males, they are the what? For the gonads? Testes. For the ovaries, they make the oocyte or the ovum. Okay, for the female gamete. And for the males, and then the hormones are that the ovaries produce are estrogen and progesterone. And for men, they have the testes. And once again, their male gamete is the sperm. Very good. And the testosterone that's the testes produce are called the testosterone. Very good. Okay. Clitoris is this structure over here. For the males is the glans penis. Those are homologous structures. They're both highly sensitive and also erectile tissue, so they can become aroused when it becomes erect when it's aroused. The labia majora is these structures on either sides of the vagina here, and the scrotum are homologous to each other. Okay, and they protect and cover the reproductive structures. The vestibular glands for the females, there's no picture for it per se, but they are located on either side of the vaginal canal. But it's equivalent to the bulbourethral gland for the males. They secrete mucin to lubricate. For the vagina, it lubricates that. For the males, it helps provide some secretion and part of the seminal fluids. When we look at the perineum, it's this diamond-shaped structure that we have between the pubis and the coccyx. The urogenital triangle is the anterior triangle that you see here of the perineum. For the females, it contains the clitoris, the urethral orifice, and also the vagina. For males, it's just the base of the penis and the scrotum. So base of the penis is over here and parts of the scrotum over there. The anal triangle is the posterior triangle of the perineum, and it contains the anus itself. And the perineal muscles we talked about in the past before, so some of this should be kind of a review for you. We have the ischial cavernosus. The ischial cavernosus is on either side here. Okay. And then the bulba spongiosus is this medial muscle here. And then we have the superficial transverse perineus on either side here. Then there's a middle layer, which the deep transverse perineus, and the external anal sphincter. And there's also an external urethral sphincter, which we already talked about, right? when we were talking about the urinary system, right? And where is it located again? In the, the urethral sphincter, external urethral sphincter, in the urogenital diaphragm. Very good. The innermost layer has the, what do you call this muscle here? Coccygeus, right? And then you have collectively over here the levator ani, right? With the levator ani, you have the one in the anterior side, the pubococcygeus, and then the one that's a little posterior behind that is the ileococcygeus. So all this kind of comes back, right, from the muscular system. Okay, primary sex organ for the females, what is it again? Sorry? The ovary, right? The ovary, very good. Then it has a lot of these accessory sex organs, which we'll go into next. Okay, so let's first talk about the ovary itself. Surrounding the ovary is a fibrous capsule called the tunica albuginea. They have different regions to it. It has an ovarian cortex, which you see over here is the outer parts here. And what do you think the inner part is going to be called? Yeah, the ovarian medulla. The ovarian medulla has the blood vessels going into there, nerves and lymphatic vessels. Within the ovarian cortex over here are the follicles, and we'll talk about the different follicles in just a second, so don't worry about that for now. Okay, and before we go any further, let's first talk about mitosis versus meiosis. Mitosis we went over back in Unit 1 already, so let's talk about meiosis now. So meiosis is similar to mitosis in that it has prophase, metaphase, anaphase, and telophase, but it goes through two cellular division. At the end of mitosis, right, what do we have at the end of mitosis? Two daughter cells, right? And the two daughter cells are identical to the parent cell, okay? However, for meiosis, they're going to end up having 4 different types of cells, okay? And these cells are haploid cells. Haploid meaning we only have half the amount of chromosomes, okay? Half the chromosomes. How many chromosomes does a human have? 46 chromosomes total. Okay, you could also say that there's 23 pairs of chromosomes. Does that make sense? 23 pairs of chromosomes? Because one set comes from our dad side, and the other set is coming from our maternal side. Okay, so that's why there's 23 pairs. Diploid cells are these 23 pairs. So when you see diploid, the di means that there's a pair of those chromosomes. While haploid is just one set. Okay, they're not paired off. And this makes sense, right? Because when you have a sperm, how many chromosomes are in a sperm then? Is it haploid or diploid? Haploid. There's only 23, right? And in an egg, there's how many? Only 23 too, right? So when you have a zygote is when the sperm fuses with the egg and it's a fertilized egg. How many total do you have now? 46, which will become a human afterwards or after development, right? So here you have mitosis. Mitosis produces 2 identical diploid cells, while meiosis, we said how many cells do we produce? 4 cells, right? But what kind of cells are they? Haploid, okay? So it's a difference in that. Okay. So of course they have an interphase. The interphase once again has G1 phase. The G1 phase is just a period of time for cellular growth. And at this point in time, the centrioles replicate. What is the next phase again? Do you guys remember? S phase. What does S phase do again? Yeah, replicates the DNA. Okay, it replicates the DNA. And then the G2 phase is continual growth. At this point in time, the beginnings of the chromatin starts to condense, okay? But not quite, right? Because most of the condensation occurs where? In what stage? For a chromatin to condense into chromosomes? Mostly in what stage? Prophase, in prophase. Okay, so that's interphase. Meiosis is different from interphase. So the very first stage of meiosis is prophase 1. And prophase 1, just think of prophase in mitosis. What will be occurring? What's happening to the nuclear envelope? Dissipates. Okay, disappears. Good. Nucleolus? Disappears too. Good. The chromosomes start to form, right? Starts to condense, right? Kind of thing. Very good. So all those are happening. And the other thing that occurs in prophase 1 that's a little bit different is that the pairs that are homologous, they start to pair up and forming tetrads, okay? The forming of homologous, what are homologous pairs? Remember how we said that one set of chromosomes comes from our mother, right? And the other sets comes from our father, okay? So the mother and father chromosomes, just say this is chromosome number 1, okay? They will pair up together to form this tetrad over here, okay? If there was chromosome number 2, it will be same thing, so on and so forth, okay? So they'll all pair up with each other. And the purpose of this pairing up allows them to do this process called crossing over, okay? Meaning that they're going to exchange parts of the mother's chromosomes to exchange it with the father's chromosomes because they're so closely together that some of the chromosomes will start to swap with each other, okay? So let me just redraw this real quick. So if you think about, okay, this is the maternal chromosome. Let's just say that this is the paternal one. Okay, so they kind of overlap each other. Okay, and when they overlap each other, they switch off parts of their chromosome at the same location. Okay, and I'm going to just redraw it now to assume that they kind of swapped off parts of their chromosome here. So now it'll look something like this. Okay, before they were together, they would just look like this. Yeah, and they form a tetrad over here, and then if you were to separate them, they'll look like that. Does that make sense? What crossing over is occurring? And the thing about this of what's happening is that we're increasing the amount of genetic diversity. What I mean by that is that the chromosome that we just formed now, it's neither like our mom or our father's chromosomes now, okay? Because our mom's chromosome looked exactly like this. Our father's chromosome looks exactly like this, right? Kind of thing. But when you have crossing over, now you have like a combination of the two. And so it increases the amount of diversity in our genes now, in the gene pool, when we're talking about the population and stuff like that. Okay, so it makes a little more difference. And the importance of that in an evolutionary standpoint is that if some kind of environmental change occurred, hopefully someone has a genetic makeup to survive that change in the environment in order to continue on with the species. Okay, so that's prophase 1. What's going to happen in metaphase 1? What happens in metaphase in mitosis? Yeah, they line up at the metaphase plate, right? So same thing. The chromosomes are lined up at the metaphase plate. What else is happening here? The spindle fibers, they're going to attach to the centromeres. Remember those proteins around the centromeres? Those are called kinetochores. Yeah, So the spindle fiber attaches to those proteins called kinetochores at the centromeres. Okay. Next is anaphase 1. What happens in the anaphase in mitosis? Yeah, the chromosomes are separated, right? Kind of thing. And they're kind of like basically going to either side of the poles, right? Similar to that, but different, okay? The chromosomes are actually not separating themselves. Remember how we said it was a tetrad, right? Tetrad meaning that it was one from our mom and one from our dad side by side to each other. Now they're going to be pulled apart, okay? They're going to be pulled apart. So our mom's chromosomes are going to be pulled to one side, our dad's chromosome is going to be pulled on the other side. But in what order are they pulled is not determined. So that's why it's called random assortment, okay? What I mean by that is like, just say, where's my, okay, here. Okay, just say, I'm going to just say there's 3 chromosomes like this, okay? This person only has a total of 6 chromosomes total, okay? So that's the 3 pairs of chromosomes, okay? So random assortment means that, okay, potentially I could pull these 3 chromosomes to become that new cell and these 3 chromosomes to become the other new cell, right? Kind of thing. So at the end, you'll have a cell with these chromosomes and then you have another cell with these chromosomes, okay? That could be one outcome of it, right? The other outcome of it is that just say since it's randomly pulling it, let's just pull this one here to this side and this one to this side, but I pulled this one here over, okay? So instead, I end up having this cell instead, okay? You'll end up having that cell on that side and this cell on this side. Okay, and you notice how this cell is different from this cell here, right? And this cell is different from that cell, right? So this is this random assortment that occurs, meaning that the way that the chromosomes line up before it gets pulled apart will cause this random assortment to occur. Okay, once again, you're increasing the diversity of the gene pool again, because no one will have the same set of chromosomes since you're randomly pulling them apart with each other. Okay. Telophase 1. What happens in telophase? Chromosomes decondense. The nucleus reappears. Nuclear membrane reappears, yeah. What else reappears? Nucleolus reappears, good. What happens to the spindle fibers? Dissipates, right? And we already talked about the chromosomes also decondensing, right? Decondenses into what again? Chromatin. Okay, good. So all that occurs, right? And what happens to what do you call that pinching off again? Cytokinesis, yeah. Cytokinesis also finishes too, right? And at the end, at this point, we have haploid cells. All right, 2 haploid cells. Okay, now we enter prophase 2. Okay, what happens in prophase? Just what happened in general in prophase. Okay, chromosome starts to condense again. nuclear membrane envelope dissolves, nucleolus dissolves, spindle fibers start to form. All those occurred. So same things, right? Centrioles move to the poles. So far everything is the same. Actually, meiosis 2 is actually very similar to regular mitosis. So at metaphase 2, we have the chromosomes lining up at the metaphase plate. Very good. And then anaphase, okay, spindle fibers attached to the kinetic cores, right? Anaphase 2, we have the chromosomes. What's going to happen in anaphase 2? They're pulled apart. Okay, in this case scenario, the sister chromatids, right, are going to be now daughter chromosomes, right? So they're going to be split, and the chromatids now go to the opposite poles. They're now daughter chromosomes. And telophase 2, what's going to happen? Chromosomes decondense, nuclear envelope reforms, nucleolus reforms, spindle fibers dissipates. Very good. All those occur. Cytokinesis also completes, right? And at this point, we have now 4, right? Because originally we had, in meiosis 1, we have 1 cell becoming 2 haploid cells. And then from one of these haploid cells, they will undergo meiosis 2, forming 2. And this one will also undergo meiosis 2, forming 2 more. So we have a total of 4 haploid cells at the end. Okay. The reason why I wanted to go through meiosis first is because it will make more sense of what oogenesis is about. Oogenesis is how we produce the ovum. And it's probably a little bit more complicated than spermatogenesis because the oogenesis, they get halted at different stages. The oogonium is the stem cell for the females to produce the different oocytes. So within the female embryo, it undergoes mitosis, okay? So this one always undergoes mitosis. When it undergoes mitosis, okay, oogonium undergoes mitosis, so it makes 2 cells. Are these 2 cells identical? Yes, because it undergoes mitosis, right? So it will be 2 same cells, okay? You make a few thousand of these during the first few months of development. Okay, so you have the mitosis occurring. The thing is that one of these will remain an oogonium, while the other one will become this primary oocyte. Okay, this primary oocyte will undergo meiosis. This oogonium will undergo what? Mitosis again, to form, once again, 2 identical cells, one continue to be the oogonium, the other being primary oocyte. Does that make sense? Okay. So these cells are diploid cells. There are 23 pairs of chromosomes. The primary oocyte, you have about 67 million of these when they are developed, but when you're at birth, for females, when they're at birth, they only have about 1 million left. However, once puberty starts to occur, they even have fewer at that time. Okay, so you notice how it just gets fewer and fewer over time. So over here, they are rested at prophase 1, meaning that at birth, pretty much, they stop at prophase 1. Okay, so before birth, they have 6 to 7 million. At birth, about 1 million are left, and they stop at prophase 1. A lot of the ones that they used to have 6 to 7 million but only have 1 million, All the other ones underwent this process called atresia, meaning degeneration. We're still at the diploid stage here because it hasn't undergone meiosis yet, right? It's stopped at prophase 1, right? It's not only, when does it become haploid cells? At the end of meiosis 1 or 2. 1. At the end of meiosis 1. So at puberty, like I mentioned, there's even fewer primary oocytes. There's about 250,000 primary oocytes. And one of them will end up completing meiosis 1, okay? For each month. That one that undergoes meiosis 1 will be the secondary oocyte. It is the larger of the two daughter cells. The other cell that's produced after meiosis 1 is called a polar body. Okay, so we have the primary oocyte that we just talked about. So it's going to undergo meiosis 1 to form a secondary oocyte and a polar body. The polar body is basically, it gave up all its cytoplasm, all its organelles and everything so that it could help sustain the secondary oocyte. So it's just basically whatever leftover remnants of that cell. Most of the cell materials went into the secondary oocyte, while the polar body is just the remnants of it. This will be stopped at metaphase 2. So we stop at metaphase 2. At this point in time, after meiosis 1, we have a haploid cell, 23 chromosome. Like I said, the first polar body is just the smaller of the two cells here. It possibly can go through meiosis 2, but it could also degenerate as well. Okay, so we wouldn't need to worry about that one too much. Okay, the secondary oocyte, it will undergo meiosis again. It will complete meiosis 2 only after fertilization occurs. Meaning that once the sperm starts to enter the secondary oocyte, it's going to finish up meiosis 2 to form an ovum and also another polar body. Once again, this is to end up having most of the cytoplasm and all its cellular material into the ovum. Why do we want to put all this material into the ovum for? Why? How come they end up forming these polar body? Why not equal size cells? No clue. Okay, so eventually this oma will become what? Yeah, a kid, potentially, right? A kid eventually, kind of thing. So if they were just equal size, right, then you're not giving it enough organelles to begin off this process of development and stuff like that. So you want to have most of the materials into one cell so that it will be easier, more efficient for the cell divisions to occur and stuff to end up forming a child at the very end, kind of thing. So that's the reason why. You notice that at this period of time, there's only 400 to 500 ovums that are produced. Okay, I guess technically speaking, we should be saying that 400 to 500 secondary oocytes are produced. Okay, approximate. Because the ovum It depends on whether or not it's fertilized, right? If it's fertilized, then it could be even fewer than 400 and 500, right? Kind of thing. So the secondary oocytes should be the 400 to 500. And how do you get this number? Just think about it. How often does a female ovulate? Approximately once a month, right? Okay, how many months are there in a year? 12. Okay, when do they become sexually mature to start to potentially have a child? Around 12 or 14, right? Just say around 12, just say. Okay, so if we round it down, it'd be around the age of 10. Okay, 10 is a bit young, but I'm just doing a round number to make the math a little bit more simple. When does menopause usually occur? Around like 50-ish kind of thing, all right? 65 is a little too old. Around 50, so that's about 50 minus 10, about 40 years-ish, right, kind of thing. So if you multiply these two numbers here, right, then you have about 480, so there's this 400 to 500 number that you got, okay? So approximately. All right, so once again, the ovum's 23 chromosomes, and then we have the secondary polar body we just talked about, right? That forms next to the ovum.
So does menopause occurs around the same time that people get people need to use reading glasses?
Sure. I think that's a little bit different. It's just that for that, it's that the ligaments, remember the suspensory ligaments that we talked about, they don't function as well because the lens itself starts to harden, so it's not as pliable, so it stays the same shape. It stays the same shape. Okay, once the sperm unites with the ovum and fertilizes it, then we have something called a zygote. And how many chromosomes is a zygote? 46. Very good. All right, let's talk about the ovarian follicles. Where do you find the ovarian follicles again? What part of the ovary? At the cortex. Yeah, very good. At the cortex. So the ovarian follicle has the oocyte or the oogonium, depending on which part of development we're talking about. And around the oocyte is the granulosa cells. These granulosa cells help produce and support the oocyte. Its main function also is to produce estradiol. So all these cells that you see around the oocyte are granulosa cells. And surrounding the granulosa cells, then we have something called the theca cells. Do you guys remember the theca cells and what it produces again? When we were talking about it in the endocrine system? What do females need in order to produce estradiol? Say that again? Estradiol is a type of estrogen. So how does it produces that estrogen again? Okay, yes, it needs that in order for it to produce estrogen, correct? But in order to produce the estrogen itself, it needs an androgen, right? And the theca cells, they are the ones that make the androgen and then transport it to the granulosa cells to make estradiol. Okay, it may or it may not be present, and we'll talk about that in just a second. Okay, folliculogenesis, this is developing the follicle itself. We start off with the primordial follicle. Okay, the primordial follicles usually occur at the very edge of the cortex itself. First off, what you have here on this picture is not reality, meaning that you don't see the development of the follicles going in this direction, okay? It does not occur like that in the real ovary. They did it so that they don't have to redraw the same pictures over and over again. This is as is assuming that we're just looking at a imaginary ovary, that you could see all the different stages of the follicles developing at the same time. Okay, but it's, in real life, it's not going to look like this. Okay, so but anyways, the primordial follicles you tend to see near the edge of the cortex here. Remember, this is if you're looking at the primary follicle, this usually forms prior to birth, but it remains there after birth. Okay, these primordial follicles, and most of these would be primordial follicles when you're born, and it's not until you hit puberty that these start to develop into like the primary follicle, secondary follicle, so on and so forth. Okay, at this point in time, we have a primary oocyte. Why is it primary oocyte and not an oogonium? Okay, good. It's the meiosis 1 stage, and what other thing is also occurring? What did we say the oogonium, when did it stop? Or when did we have no more oogoniums again? After birth. So after birth, no more stem cells, basically. No more oogoniums. Okay? So that's why females have a limited amount of eggs that they will end up having. Okay, so that's why we have a primary oocyte at this point in time. The primary oocyte has only a single layer of these granulosa cells. They tend to be very flat, so they're squamous in shape. Okay, so this is the primordial follicle here. When it starts to develop a little bit more, those granulosa cells around the primary oocyte is now cuboidal in shape. Okay, so this is a primary follicle. A secondary follicle starts to develop once, like we said, puberty occurs, and now we're developing into the primary follicle to a secondary follicle. And at this point in time, you notice that the primary oocyte is still located here, but you notice how it's not one layer of granulosa cells, but multiple layers now of granulosa cells. Okay. Potentially, what's happening is that an antrum will start to develop. This one doesn't have an antrum that you can actually see, but antrum is this fluid-filled space that you see, like in this structure over here. There's a fluid-filled space over here. That's an antrum starting to develop. Around the granulosa cells, we start to have the theca cells starts to develop now. Okay, theca cells starts to develop around the granulosa cells. Potentially, you could see something called a zona pellucida. It's a glycoprotein gel that is around the oocyte. Let me erase that. Okay, so you do see this as well. Here's the zona pellucida around the primary oocyte here. Once again, what do you call these cells here? These are the... Oh, on the bottom right-hand corner. Sorry, my bad. Bottom right-hand corner has the primary oocyte, the zona pellucida around it, and what do you call these cells that I'm kind of shading in right now? Still not working? Oh, it's on the bottom right-hand corner. Sorry, that's why. Okay, once again, what do you call this cell here? Primary oocyte. Primary oocyte. Surrounding that is this glycoprotein here, zona pellucida, and these cells that are surrounding the oocyte is the no. This is the granulosa cells. Okay, the theca cells is around that. Okay, these cells that are around the granulosa cells are the theca cells. Let me redraw the theca cells for you. So this is the theca cells. The theca cells could be potentially two layers. There's a theca intima and a theca externa. We have the pre-ovulatory, or also known as the graphia, also known as the mature follicle. And what do you notice from a mature follicle? Clearly you can start to see that the antrum is a lot larger. Okay. And it's at this time that meiosis 1 finishes. Okay. This is where the secondary oocyte is now occurring. And we have the theca externa on the outside and the theca interna. Okay. It's the theca interna that makes the androgen called androstenedione that is transported to the granulosa cell. The cumulus oophorus is the granulosa cells that are surrounding the oocyte itself. So these cells here are the cumulus oophorus. What do you call the glycoprotein around the secondary oocyte again? Zona pellucida, right again. And the innermost layer of the cumulus oophorus is the corona radiata. So just this innermost granulosa cell layer. After ovulation occurs, so the secondary oocyte is now gone, the remaining of the cells that remain, the granulosa cells, theca cells, so on and so forth, they will end up developing into something called the corpus luteum here. The corpus luteum functions to produce a lot of progesterone, and what progesterone will end up doing will cause the endometrial lining to continue to maintain the endometrial lining. It makes sense because potentially when you have a fertilized egg, you want the fertilized egg to implant itself on the endometrium. Eventually, if there is no implantation of the embryo, you have a corpus albicans that develops. The corpus albicans is the degeneration or scar tissue that occurs of the corpus luteum. So if there's no pregnancy, a corpus albicans occur. Here we have most likely like a cat ovary. The reason why you could tell is by looking at how many mature follicles that we have, or pre-ovulatory follicles that we have. If there's multiple pre-ovulatory follicles, we know, okay, this cat is ovulating multiple oocytes at the same time. And that's why they produce litter kind of thing, versus for humans, we only produce one, about once a month. All right, so looking at this, what do you call these cells here, or these follicles here? Not the primary oocytes, but the... No. Primordial follicles. The primordial follicles, what do you call one of these cells here? What's one of these cells? Okay, say again. Not oligonium, right? Because oligonium occurs prior to birth, right? No. So instead of an oligonium, you have a primary oocyte. Okay, that's the primary oocyte. These cells around it, which we don't have it zoomed up enough, but the cells around the primary oocyte you have as the granulosa cells. Okay, what shape are these granulosa cells? Yeah, squamous, right? It's only one layer, simple squamous. Okay, as this continues to develop, it'll look something like this here. What do you call this structure here? What was this one here? One of these? These are called primordial follicles, right? Here you have the primary follicle. Okay, you have the oocyte here. What kind of oocyte is it again? Primary oocyte. And you notice how the cells that are surrounding it, what kind of cells are these again? Granulosa cells. And notice how the shape of it is different now. It's a little bit more cuboidal in shape. Okay, how many layers are there? Just one. Very good. Okay, so looking at this here, what do we have now? Secondary follicle, right? Secondary follicle. What do you call this cell in the middle here? Still primary oocyte, okay? And all these cells that are surrounding it? Granulosa cells, how many layers now? Multiple layers, okay? And potentially, you don't see it too much on this one, but potentially on some of them you could start seeing the theca cells surrounding it, okay? What do you call this that starts to develop too? Antrum. It's a fluid-filled space. So here is a more developed secondary follicle here, okay? Okay. What do you call this one here then? Just wondering. No, not yet. Look at the antrum here. There is an antrum, correct? How big is the antrum? Not too big. So what is it then? Secondary follicle. Okay. How about this one here? Secondary follicle, the antrum, not that big, right? Kind of thing. What would you call this one here? At this point, I would still call it secondary. Primary is only just one, like, layer of cells, right? Kind of thing. Okay, how about this one here? Okay, in comparison to the other secondary, the other secondary follicles that we just looked at, right? Right, this is a secondary, right? This is a secondary, here's a secondary. How big is the antrum on this one?
So that would be mature.
Okay, this is the mature or pre-ovulatory follicle. Okay, what kind of cell do we have here? Okay, now it's a secondary oocyte. At this point, meiosis 1 finished, okay? So we have the secondary oocyte. What do you call these cells around here? Granulosa cells. What do you call this glycoprotein around here? Zona pellucida. How about this, the granulosa cells specifically, or just this area here? Corona radiata. Good. Part of the corona radiata actually is just around the granulosa cells around the zona pellucida. But what do you call everything over here? Cumulus ovoris. And then these cells around everything else here. What are these cells here? Theca cells. Very good. All right. On this, you don't actually see a corpus luteum, but they're pretty obvious if you were to take a look at them. All right, so let's look into the ligaments now. They help suspend either the ovary or the uterus itself. So there are ligaments that suspends the ovary. There's the broad ligament. The broad ligament is basically majority of the ligaments here. So this is all broad ligament that you see here. Okay. This is all part of the broad ligament. The broad ligament is composed of multiple smaller parts. So if we're talking about the mesoovarium here, this is the one that anteriorly holds the ovary to the broad ligament here. So if we look at it, here's the ovary. So this is the mesoovarium. part of the broad ligament. The mesosalpinx is the superior part of the broad ligament that holds the uterine tube to the broad ligament. So here's the uterine tube over here. Here's the broad ligament. So this is the mesosalpinx over here. Okay, let me just, I guess, show in different colors. So here's the mesoovarium here. Mesosalpinx over here. And the mesometrium is a part of the broad ligament that holds the uterus to the broad ligament. So here is the mesometrium. Right, all of them are part of the broad ligament though. The ovarian ligament, like the name implies, will attach the ovary to the uterus. So what you have here is the ovarian ligament here. Suspensory ligament has blood vessels and nerves going through it and attaches the ovary to the lateral side of the pelvic wall. Okay, so what was this structure again? Ovarian ligament, right? This is the suspensory ligament over here. Okay. It once again attaches the ovary to the lateral walls of the pelvis. Okay, for the ligaments suspending the uterus, we have the round ligament that we just talked about. No, sorry, not the round ligament. We have the broad ligament that we talked about earlier. The round ligament is this ligament here that ends up attaching the uterus from the lateral sides to the labia majora. What was the labia majora again? Do you guys remember? That was on the outer surface, yeah, in the perineum area. And goes through the inguinal canal. And then you have something called the uterosacral ligament. And like the name implies, it's going to attach the uterus to the sacrum. Very good. And you can see the uterosacral ligament over here. This structure over here. Okay, so you see this? What is this attaching to? That's the ovarian ligament. Very good. Attaches the ovary to your uterus. Very good. This doesn't actually show it, but if you have something attaching it from this direction, what do you call that then? Suspensory ligament. Very good. The round ligament, you'll see somewhere around this area, kind of thing. Okay, the uterine tubes. So when the ovulation occurs, the ovum will end up going, or the secondary oocyte will end up going into the uterine tubes. They are also known as the fallopian tubes or the oviducts. They contain ciliated simple columnar epithelium, and ciliated so it could help propel the oocyte into the uterus. There's multiple parts to them, to the uterine tube. The very first part is called the infundibulum. On the infundibulum, there's little hair-like structures here. These are called fimbriae. The fimbriae are the ones that will end up flapping so that it draws the oocyte into the uterine tube. The ampulla is next. It's this enlarged area here of the uterine tube. And then finally, you have the isthmus over here. Where fertilization usually occurs, it usually occurs at the ampulla area. So when the sperm ends up going into the female reproductive tract, it will travel all the way to the ampulla over here. All right, let's take a little break before, I guess we could stop here, or do you guys want to? I mean, regardless, we're going to take a break, but I don't know if we want to continue on with this lecture or or look at the lab stuff. Who wants to continue? Just wondering. Okay, who wants to actually do lab stuff? Okay. Well, we'll just continue, but let's take a break for now. We'll just continue on with the lecture, though. Okay.